Welcome, everybody. Good morning. And thank you for attending this second day. We're very excited to have you here, and we have a really wonderful session for you this morning and as well as after this. So what I want to talk about today, and I'm the moderator for the next three speakers, we want to talk about a population that isn't human and a population that aren't children. So why are they important to us? Well, these are important because these are the microbiomes, the bacteria and the viruses that live around us, that probably live within us, most of these. And the people who have been studying these, in fact, some of the people that we're talking about today, have done groundbreaking work in the area of understanding how these organisms live with us, live within us, live outside of us, and how they affect maternal and fetal health and the life course of the human being. And so to start off, I'd like to introduce Dr. David Relman, who's the Merrigan Professor of Medicine and Microbiology and Immunology. And he is one of the pioneers in the area of the discovery and the definition of the human microbiome, using his advanced molecular techniques over the many years that he's been here at Stanford. And so thank you very much, David.
It's a pleasure. So it's a pleasure and honor to join you here this morning. Bonnie already introduced the concept that much of us, in fact, is not of eukaryotic origin. And so that's going to be the theme of much of what I will talk about, and in fact, the subsequent two speakers. The topics I thought might be of interest and relevance here are premised on concepts that ecologists have spoken about and studied for quite some time. And for that reason, I thought I would talk about ecological landscape as a nice starting place. Ecologists also were quite interested in the role of history in determining the subsequent structure and functioning of the microbial communities that they study, and in fact all communities that one might study. And so that too is something that I want to touch upon because in fact we are very much about our history as well. Third, much of life is about the exposure to and recovery from disturbance. And I thought I would introduce that concept in the context of pregnancy and think about with you whether pregnancy ought to be viewed as a form of disturbance to which we have, of course, adapted over quite some time, but the implications of thinking that way about this other component of humanness, and then leave you with some thoughts about what are the pending unanswered questions in this area of work. When one thinks about states of stability in a complex system, many have been drawn to this imagery of the topological landscape, the idea of a surface. It's drawn here in 2 dimensions, but you might think of it in 3, which has in it a number of different valleys, depressions of varying depths. Those depressions are meant to signify, in this case, a stable state, a state in which a community ecosystem may find a great deal of robustness and resistance to change. So the deeper this valley, the more stable would be the system. And you can see these systems depicted as little interactomes of networks of interacting organisms, and they're interacting with an environment as well. There are, of course, multiple stable states, and the landscape, the shape of this landscape, determines the likelihood that a given state will remain as is or be subject to change. Now, this landscape in this setting today, in these discussions, is us. And so as clinicians, one of the goals might be for us to be able to describe the landscape, the topological landscape, which is each of our patients or anyone that we seek to understand and help manage in the form of their health. And so I'll talk a little bit more about why that might be an important way of thinking about the human microbiome, the collection of of organisms that come together, have co-adapted with this host, us, and form these communities, and then the subsequent states that they occupy over time. Just to remind you, we now have a great deal of building data and evidence that suggests that the microbial communities of the human body are inextricably involved in some fundamentally important biological, physiological, clinically relevant processes that, that underlie all that we are. So ranging from digestion to nutrition to the early instruction of our immune system, the processing of foreign compounds, the xenobiotics, the management of our exposures to all sorts of day-to-day and varying kinds of change and disturbance. So this is all that we have come to know to be important about our microbiome. And needless to say, much of this might be seen as directly relevant to the time of life that I'm going to talk a little bit more about, pregnancy. Pregnancy is, of course, important as the historic backdrop, the preceding state in time that, that may, in fact, be so important in determining the nature of the microbiome that's presented to a child at birth. And yet, we think less about the microbiome of a mother when we're worried and thinking about what is the role of a microbiome in a baby and what might it bestow upon that baby in terms of future health and possibly susceptibility to disease. There are a number of ways of thinking about this history prior to birth and its relationship to the microbial communities, the microbiome. One is to think more about what are the states of maternal health during pregnancy and how might they affect the mother's microbiome, which in fact will be the microbiome that is transferred to the baby, at least in part for some period of time. What about the changes that take place in a woman at the time of conception? We know essentially nothing about the effect of conception, the onset of pregnancy, on the maternal microbiome. What about the subsequent exposures of the mother during pregnancy? Diet, drugs, intercurrent infections. These are all things we know to affect microbiomes. We've studied very little of this in the setting of pregnancy. And yet there are some important theories and concepts that would suggest that we ought to be looking carefully at these various factors, this history in the mother. This is a review that was published by a colleague here at Stanford, Tad Fukami, in the Department of Biology. Tad studies the process of community assembly. This is, as you may know, one of the fundamentally important aspects of community structure and function. That is, how did it come to be? And what Tad has reviewed in this paper and what has been demonstrated time and again in a variety of traditional ecological studies is that this history is exceedingly important in ways that you might not have thought about. For example, what is the order of arrival of these microbial colonists from the time of there being a pristine, microbe-free state? And what is then the impact of each arriving colonist on that landscape and the subsequent changing of the landscape such that the next arriving colonist now sees a slightly different landscape in front of it? These are things, again, that we know to be very important. And they ecologists, and I think we should be talking about historical contingency. What's the effect of order and timing of past events on a current complex system like the microbiome? These events could be abiotic or biotic. This is one way of classifying. Abiotic would refer to disturbances like change in the physical change in the landscape. You might think as well about changes in the chemical environment, the hormonal changes of pregnancy, and how that might reshape the landscape at the time of either rebuilding a microbiome during pregnancy or the subsequent transferal to a baby. And then biotic, the actual species and their arrival. Again, there is growing work, bodies of work, to suggest that these are not just concepts that sound plausible and relevant, but in fact can be demonstrated to be important experimentally. And this is a paper I won't review, but simply to highlight, recently published, Andrew McPherson and his colleagues in Bern, Switzerland, in an elegant series of experiments show that a mouse, when transiently, a germ-free mouse, when pregnant and then transiently exposed to microbial species, but transient in the way that they then disappear before delivery, that the subsequent pup, this is a mouse pup, now has a different looking immune system, a different trajectory of immune system development, and a whole set of different associated properties, physiologic properties, than would a mouse that was born to a germ-free mom that had none of those intrapartum during pregnancy exposures. So we need to be thinking now about this history and its impact on fetal development, fetal health. We know about epigenetic imprinting. I would submit that the presence of an evolving microbiome during pregnancy is another form of both direct and indirect epigenetic imprinting that's now going to be transferred to a baby and play out over subsequent years. And this idea of playing out over time is something, again, that's been hypothesized to be important, the idea of serial successions of community propagation and assembly. This, again, is not just a concept, but has been shown in experimental work, again, now here at Stanford by Justin Sonnenberg, in a really interesting series of experiments where he altered the diet of a first-generation set of pregnant mice and then looked at subsequent generations of mouse pups born to a mother that had been put back on an originally, in this case, fiber-deficient diet, and saw that it was only over successive generations that one saw the persistence of an effect on these growing intergenerational mouse pup microbiomes that he then concluded was the result of this history that had now been propagated out over time, now over generations. So what about the effects of pregnancy on the microbiome? There are some data in the literature. We saw it here in work that was supported by the March of Dimes Foundation and through a prematurity research center that I think you may have heard about, hosted and guided by some of the people in this room. We saw it as part of a microbiome-oriented component to look at what happens to this microbiome during the course of pregnancy. And I won't belabor the details. This is published work, that which I will show here. But in short, we looked at a variety of body sites in a mother, looked at this longitudinally over time, and asked the mother to sample once a week during the course of pregnancy, although this begins, generally speaking, only after 12, 14, 16 weeks of gestation, because we had the challenge of trying to get to women who were now pregnant and willing to join our study. And I'll tell you about some of the limitations at the end. So this was sort of the setup, weekly sampling, these little green bars, multiple body sites, lots of samples, collection of mother host-related samples as well. Well, for the purpose of host-related analyses, everything is a host, an environment. And I'll tell you about, again, the data that were published for a set of 49 subjects. We started with a preliminary set of 40 and then sought to validate some findings in the subsequent 9. I will simply say that these were women recruited from the Palo Alto area, so they had reasonably good representation within the Hispanic ethnic class, but limited representation within various racial classes, including African Americans. And the women who had delivered prematurely, which in fact is one of the major goals of this research center, to understand the basis for prematurity, these women in our study delivered generally a bit late for a premature delivery. So again, one of the limitations was the limited number of subjects that we had, their restricted racial background, and the numbers of women in particular who had delivered prematurely, given that this was one of our primary goals to understand. So lots of samples over time. These are the 4 body sites. Each of those little rows is a woman, and the dots are their samples. You can see by the black line, which is time of delivery, that we continued collection. This research center, and all of its wonderful staff continued the collection of samples after delivery, and I'll tell you a little bit about, in one slide, those data as well. This is a display using a form of ordination, of course, premised on the idea that we have multidimensional data and we're seeking to visualize it in a way that we can begin to build hypotheses. These two axes here represent the greatest amount of variation in these data, and these data are the taxonomic composition of the samples from each of these 4 body sites. And in this display, those samples are color-coded based upon the body site from which they were derived in these various 40 women now. This is the initial set of 40. So you see somewhat distinct clustering, and this based upon color, based upon body site, and this is something that we have already known to be an important premise, an important guiding factor in understanding the nature of the human microbiome. The body site is most important. Your mouth is much more similar in terms of its microbial community to the mouth of the person next to you, even though if you don't know them, than it is to your own gut, okay? So body site trumps individual. But when we talk about one body site, now individual is an important source of variation. I'll tell a little bit about that. So we see the same thing during pregnancy. We also see this body site coherence play out over time. Now the x-axis is gestational day and then subsequently postpartum day. Now, we looked in particular at each body site alone because, as I just mentioned, this huge additional source of variation, site to site, means that we might miss some of the nuances and important detail that one would see when focusing on one site. So these are the data from just the vagina of that initial 40 subjects. We have close to 1,000 samples from these 40 subjects because, again, they're weekly. And now, again, they're displayed against time, which is the x-axis. Now the color coding is based upon whether that woman delivered at term, that's in blue, or preterm, prior to the end of 37 weeks. Those samples are in purple. And what struck us was the fact that an unusually appearing large fraction of these samples from women that delivered early seemed to be of a different compositional type. And the way that you see that here is because these purple samples seem to be higher on the x-axis, on the y-axis. The y-axis here is the score that came from the first component of that principal components analysis. And that score is an indirect and somewhat crude reflection of taxonomic composition. They have a higher score, a different composition. And the question was, what might be the nature of this somewhat distinct vaginal community that seems to appear early in pregnancy and be associated with those women who will subsequently deliver early? We looked at the structure of all of the vaginal communities and found something at first which was similar to studies that have been published of non-pregnant women, which is the existence of very discrete compositional states. In the vagina, they are numbered 1 to 5, and we think, in fact, that there may be probably a larger number in a broader population of women, but the literature and our data would support the existence of discrete types of a small number. The type that happened to be associated with that early gestational picture in women who delivered prematurely is very similar to what you might be able to see on these slides as the type 4 state. So it's in that little pinkish salmon color on that top bar that goes across the top. The columns that lie underneath the number 4 are the various taxa that make up the various samples that fall within class 4, and they're little tiny columns within that segment. So what is a type 4 community? It turns out to be a community that's very distinct from all the other 4 types. It's the one type where you don't have a dominant lactobacillus species. In all other typical states of the vaginal microbiota, there is a dominant lactobacillus species, and that species may vary. Each woman here tends to have one of those types, and it tends to be maintained over time, although it's been shown quite clearly that the composition will change as a function of the menstrual cycle, for example. So we came into this study thinking, would in fact the absence of the menstrual cycle mean that there was now less variability over time, or might it mean that instead we have a different kind of dynamic of hormonal change that might cause there to be a clear effect of pregnancy of a more sustained press-like disturbance type? So we first pursued this idea of type 4 association and looked at stability as well. And here are just some data to show that, in fact, this type 4 is statistically significantly associated with preterm deliveries. And so we went to try to build a case for causation. So what do you do when you've got a putative causative factor, this atypical vaginal community, and you want to, in the future, prove causation? Well, you can't take this entity and introduce it into a new relevant host and reproduce the phenotype the way Robert Koch would have you do. And so we're now relying upon alternative postulates that have been established by epidemiologists for quite some time, looking at the role of cancer and environmental factors on the occurrence of cancer. So on the left, you see what might be akin to a dose effect. This is something that epidemiologists have typically sought to establish as one feature of a causal argument. And without going into the details, again, this is published, there is some evidence of a dose-like effect. On the right is a temporal effect. Does this putative factor appear prior to the appearance of the endpoint of interest, premature labor? And in fact, it does. Now, these are just two arguments towards a body of would-be evidence that we would use to try to assert causation. But we know that causation of prematurity is almost certainly to be a multifactorial process. We don't seek or expect to explain a large fraction of it simply based upon an altered microbiota feature. And I've talked to you about taxonomic composition. We're busy now looking at the genomic composition of these communities, both vaginal and otherwise. We're looking at the metabolite composition of these samples from all of these body sites. And yet all of that may be only one piece of what is an interwoven, inextricably interconnected set of causal factors leading to premature labor and delivery. So this is still an investigation and a pursuit in progress. Many others are pursuing the same issue. I don't have the data here to show you, but this type 4 and other type community feature is relatively stable during the course of pregnancy compared to data from others in non-pregnant times. And we're collecting now direct comparison subjects for non-pregnancy states. But it raises a series of questions. How might this be related to prematurity, for example? Well, you might think about this history of aberrant assembly and now the establishment of an altered state which has properties that might be pro-inflammatory, because we know that a sustained or aberrant or improperly located inflammatory signal may be an important trigger for premature labor. Perhaps the problem is that these altered vaginal communities are now poor, impaired in their ability to do what normal human microbial communities do, which is to exclude invading pathogens. Maybe that's the issue. Maybe it's the absence of something, in fact, rather than the presence of something that's important. We know, and I just told you, that it's, in fact, the absence of lactobacillus that helps define the most gross feature of this altered community, type 4. Maybe it's an indirect effect. Maybe either a direct product, a metabolite, or a local immunologic aberrant signal is now transmitted into the systemic immune system or into the systemic circulation if it's a metabolite. Now maybe we're talking about an indirect route to the birth sac and to the tissues that are in charge of determining the onset of labor. This is something that we're pursuing and seeking to develop models. Now, I thought I'd just put up this paper because this is a paper that raised a great deal of public awareness and attention to the possibility that perhaps there are microbes themselves present before birth in the birth sac. And I just want to, without going into the detail, suggest that we're still in a position of relative uncertainty about what that might mean. For one thing, we don't know that those findings, nor our own prior, where we looked at amniotic fluid and found microbial sequence, means that there were intact microbes, or are intact microbes. And if there are, we don't know whether they are viable. And maybe viability is not the issue. Maybe there is a dormant state of these microbes that is nonetheless accompanied by subtle patterns of expression, of activity, that is important. None of this is clear to us, nor in fact is the possibility that these sequences could be in part, if not entirely in some studies, contaminant sequences. Because we're now talking about low biomass samples like the placenta or like blood during health. So there are a number of issues that need to be sorted out, and I won't talk about these now unless there are questions later. I do want to just remind you, though, that the presence of a series of disparate microbes or their DNA does not constitute a community. The ecologists, getting back to this traditional discipline that I think has so much to offer our understanding and way of thinking, a community to them is much more than a collection. And I want to emphasize that. A community is all about interactions. It's about adaptation over long periods of time, short periods of time. It's defined in the relationship to its local environment, which is us in this particular case, and to a variety of processes that sometimes are difficult to measure. So think about this as we think about the microbiome and about our relationship to the microbial world in general. Postpartum, just one snippet from this published study, and we're pursuing this as well now. We found a very dramatic disturbance that takes place, at least in the vaginal microbial communities, right at the time of delivery. And this disturbance takes place in women who give birth to a child by both C-section or vaginal delivery. So it's not just the process of physical delivery through the birth canal, but the process of delivery itself. And you, of course, know well that there are, in either scenario, very distinct changes in hormonal environment. So maybe that's the issue, and yet it looks as though there's a clear time to restoration of what we might view as a more typical vaginal community, in this case, after delivery. You see these blue slopes with time, postpartum, trending back towards a state that I'm not showing you here, which is a more typical, normal vaginal community state. So what does that mean to have a disturbance right at the time of delivery and a time to a recovery? Well, it may mean nothing. Maybe this is just an epiphenomenon of looking at taxa, and perhaps we will find when we look at function or surrogates for function, metabolites and so forth, that there won't be such a dramatic disturbance. But I think there will be. And then we might ask, well, what is the functional consequence of this for the local environment of a now postpartum mom, as well as the systemic state of a postpartum mom? And could it be, this is just speculation, that the known linkage between interpartum period, the time to the next pregnancy, and the likelihood of a subsequent premature birth has to do with this reset? Maybe if you don't allow your microbiome to reset sufficiently, a new pregnancy now has an increased chance of going down this aberrant path or having a higher risk due to its microbiome disturbance. So this is my last slide. I'm going to leave you with some summary statements and questions. I have sought to argue that this prenatal landscape is so important. There are lots of reasons for thinking about this, from those that have studied the developmental origins of health and disease in children to now ecologists, to those of us who are trying to marry the two. I've shown you some data that suggests that maybe there is a microbiota link to prematurity, but there are lots of further steps we must take. We're seeking to understand mechanism and hoping that this is all generalizable to larger numbers of women, but we will see. I've told you about a postpartum disturbance that, again, we have yet to determine as to its clinical relevance, but we're thinking could have some. And then what are the implications? You're going to hear from Phil Tarr after me talk about these events that now take place after delivery in the child. And Phil, I think, will tell you a little bit about the importance of strains and viruses. These are two things I've not talked about in the work I've shown you, and yet these are things that we too are pursuing, and I will seek to endorse as immensely important. We have to get down to a level of granularity that is technically difficult and challenging, and yet quite important. And in part because we know these to be important factors in the larger ecosystems. So the role of the microbiome in maternal health, fetal development, these are hot topics now that I think you'll hear much more about in ensuing days and weeks, and the idea of maybe the possibility of a transmitted risk. So let me just conclude here. I'll put up some names of those I wish to thank in this work, in particular the March of Dimes Prematurity Center here at Stanford, those in my lab, Dan DiGiulio in particular, whose published work I just showed you, and then all of you for your interest. Thank you.